So last lecture we talked about why we have to produce new cells, and we gave two reasons on why we have to produce new cells. Recall that one reason was that your cells get damaged. This can be possible wear and tear. Cells get damaged. And the other reason was that was growth. Today we're going to talk about how these new cells are produced. And in order to understand that, we need to understand this process called the cell cycle. So the cell cycle is actually divided into two stages: mitosis and interphase. That's where the I is, right over here. This is interphase. I for interphase. Within interphase, we have three substages. The first substage is called G1. The G stands for growth. So what actually happens in growth? Well, during growth, cells create organelles. Let's see. There we go. That's an E right there. And protein production. So what are organelles? Organelles are substructures organelle. Oops. Here we go. Sorry about that. So organelles are substructures within a living cell that have functions themselves. So what's an example of an organelle? Well, one organelle inside our cells is an organelle called a ribosome, and that is an organelle that produces proteins. Another thing that happens in G1, so we're still on G1. Let's stick here with G1. Again, the G stands for growth. Another thing that happens is that the cell doubles in size. So remember, in order for the cell to actually create an exact copy of itself, it has to make double of everything, including its size, before it can divide into two evenly sized cells. And there's also a checkpoint right before S phase. So notice if you look on this slide, there's actually a checkpoint right over here that governs whether or not the cell can continue the cell cycle and enter S phase. So passage into S phase governed by the restriction point. You can think of the restriction point as your checkpoint. On to synthesis, we'll say. And synthesis is S phase. The next phase we're going to talk about is, so far we've talked about G1, and we're still talking about what the cell actually has to do in order to fully divide into an identical cell of itself. One other thing I failed to mention in G1 is that there's actually an offshoot onto a phase called G naught. You can think of G naught as G0. And there are certain cells that go into terminal differentiation. So what does that mean? Cells that don't continue to divide. So certain cells spend an infinite amount of time in an offshoot of G1 called G naught. For example, nerve cells and muscle cells indefinitely go to G naught. So they hang out in this phase and they don't really jump back into the cell cycle and divide. Certain cells, such as liver cells, hang out in G naught, but as you all know, the liver cells do regenerate. So occasionally the liver cells actually come out of G naught into G1, and you can actually regenerate your liver cells, which allows these liver cells to actually go into G1, continue into synthesis, out into G2, and then divide into two equal cells. So we've talked about G1. The next phase we're going to talk about is a phase called S phase. So what is S phase? Well, S phase, the S stands for synthesis. So what is being synthesized? Synthesis. Well, the genetic material is copied. And we're going to talk about specifically what actually happens when the genetic material is copied in a separate video. The next phase is G2, and that's another phase of growth. And it's the final stage before any cell division can happen. Let's see. Oh. Remember, in G2, the DNA has already been duplicated. And G2 is also another point where we want to make sure there's enough organelles and cytoplasm. We'll just say organelles to make two identical cells. In this case, it's body cells. Two identical cells. So far we've talked about G1. We talked about how there's restriction points or checkpoints. Think of checkpoints as checkpoints on a highway to ensure that the cell is able to continue. In synthesis, the DNA is actually replicated. Once that DNA is replicated, we jump into G2. That's the final stage right before any cell division can occur. And then we actually end up in mitosis, which is M phase. From interphase, we think of as a phase of cell replication. Because all the organelles and the genetic material in S is getting replicated. You have to replicate your material before you can divide into two identical cells. And then once you're ready and you've replicated all of your material, we enter a phase called cell division. Cell division here, in our case, this is body cell division, right over here, is also called mitosis. And there are subphases within mitosis that we'll go over in a separate video.